Hello, everyone. I'm Kelly Eversole, the Executive Director of the International Wheat Genome Sequencing Consortium. And I'm joined here today by Ian Henderson from the University of Cambridge. And he's going to give us a talk about recombination and chromatin landscape in the wheat genome. But before we get started, I'd like to give you a brief overview of the International Wheat Genome Sequencing Consortium, or the IWGSC. So we are an international consortium with more than 3,300 members. 913 institutes and companies are involved with us, and we are in 71 countries. We have 10 financial sponsors, which make it possible for us to do our activities and try to coordinate and facilitate wheat genomic research around the world. And I want to thank them for their support, because it also makes it possible for us to do this webinar series. So after we published the reference sequence for bred wheat, we moved to what we call IWGSC 2.0. And our focus here is really to enhance our breeding through an increased understanding of the molecular basis of traits and their allelic diversity. We have a number of activities that we're continuing to do in 2022, including the IWGSC Wheat Diversity Project, where we're trying to get 8 land races sequenced at high quality so that they will represent the full breadth of wheat diversity. We're also continuing our efforts in manual and functional annotation and providing information as to how to contribute to that. And we also are expanding our collaborations with wheat genomic tool developers to try to make sure that both the small to medium enterprises as well as our academic scientists have access to the latest and greatest tools. So our next webinar will be in about a month, and it's going to be with Nikolai Adamski from John Innes Center on the cloning of the P1 locus in Triticum polonicum. And I would encourage you to join us at that time, and you can register already, and in fact register for any of our webinars that are set up for the remaining of 2022. If you will look, you have on a dashboard, you have a place for questions. I encourage you to add your questions throughout the presentation. We will not address them until the end of the presentation, but please do use the Q&A panel. Also, I would encourage you to monitor the chat panel to see messages from the organizers, as well as to see any kind of links to, sorry, just saw someone raise their hands, taken care of. Also, you can already download the handouts of the presentations, both mine and Ian's already. And then once we have the webinar, it will be placed on the YouTube channel, and you'll be able to look at it again or share it with your colleagues. So without further ado, I'd like to welcome again Ian Henderson, who's at the Department of Plant Sciences in the University of Cambridge. And he is going to talk with us today about recombination and chromatin landscapes in the wheat genome. Welcome, Ian, and I'll turn it over to you. Thank you.
Thank you. Okay, so, sharing my screen. My slides look okay?
Yes, you're good to go.
That's good. Okay, so, well, first of all, I'd like to thank Isabel and Kelly very much for the opportunity to talk to you. And as Kelly explained, the main thing I'll be talking about today is this concept of recombination, how it is in the wheat genome, and also what the relationship is with aspects of genome organization, including chromatin. So I've been interested in meiosis for many years, since high school, which is when I first learned about it. And my lab over the last 10 years has been studying it in various aspects. Now, meiosis, as you no doubt know, is a very specialized cell division found in the eukaryotes. It's very typical of the eukaryotes. And what happens is you're starting with, if you look on the left-hand side here, oops, I guess, hopefully you can see my pointer there. But we're starting on the top left with this diploid cell. It has two pairs of chromosomes, a large one and a small one, and they're colored black and white according to whether those chromosomes came from the mother or the father of this cell. And during meiosis, what occurs is that these chromosomes replicate, and then an unusual step that's very different from mitosis is that the homologs you can see here actually undergo this process of pairing. And you can see here there's lots of little ticks on those chromosomes. This is representing DNA breaks or double-strand breaks. So something very dangerous is occurring in meiosis. You're actually, breaking the genome in many, many places simultaneously. And the reason it does that is to then repair partly off the homolog to fix these double-strand breaks, and in the process creates these crossovers or exchanges between the homologs. Then at the first division, something again specific to meiosis occurs, which is that the homologs segregate. So the two sister centromeres will go to the same pole. And then there's a second division that's much more like mitosis, where the chromatids segregate. And ultimately what you end up with is this tetrad of cells. They're haploid as opposed to diploid. And you can see from the black and white coloring that through chromosome segregation and this process of recombination, everything has become mixed up or mosaic. So the kind of net effect of reproducing in this way is that you create genetic diversity. And it's, as you likely know, in plants, animals, and fungi, this kind of unique mode of reproduction is very conserved, implying that it has a kind of very general benefit. And that's been addressed extensively in the literature. And so some of the ideas are shown here from Fisher and Muller, where we have evolution proceeding in an asexual population at the top versus a sexual population beneath. And in each case, we're proceeding to this high fitness state through the acquisition of 3 mutations. Now, in a pure clonal population, those mutations have to occur sequentially to achieve this, whereas the idea with meiosis is that if your population is reproducing this way, although these mutations arise independently, they can be rapidly combined through meiosis and fusion of gametes. And so it's essentially a group selection benefit. But the idea is that being sexual allows you to adapt faster and evolve more rapidly to the environment. And so what I'm going to do in today's talk is firstly, obviously, talk about some of the work we've done in wheat, but I'll also be comparing it to a lot of analogous experiments that we've done in arabidopsis. I think it's an interesting comparison because, as we'll see, the genomes are kind of really at two ends of the scale when it comes to size within plants. But what this animation is showing is data from arabidopsis, and all this is the 5 arabidopsis chromosomes. The black circles represent the centromeres, and what's been shown here are the genotypes of sibling F2s, and the genotype key is on the right hand side there. And so what you can see is that in each of these F2s, you're getting a kind of different mosaic of patterns of inheritance of the 2 parents, designated as A or B. And so one thing that, the general thing we're interested in is, how is firstly like the number of these recombination events controlled? What controls their location along the chromosome? And how does it all play out to kind of create this kind of different mosaic of information each time you you make a new individual. But you can see again the outcome of meiosis is to create this kind of this genetic diversity. I'd also just like to quickly highlight this review article that I was part of from last year. This was kind of spearheaded by Matthew Reynolds and Peter Wittich, who some of you may know, and it's basically a kind of very broad review looking at areas where research could make a difference in terms of increasing crop productivity. And the part that Peter and I worked on was this idea of recombination and could it be changed in some way in order to kind of facilitate crop breeding or crop improvement. And this is just to kind of illustrate why sort of we consider it important to understand recombination. And so obviously in crop breeding, this is a major tool. It's classified as non-GM and it's very powerful. So for example, what we're considering here is integrating a target locus P1 from this red parent into an otherwise blue parent background, P2 background, over kind of subsequent generations of backcrosses. And so with wild type, in this cartoon, we've shown that it's achieved after 4 generations. And what we highlight are 3 possible scenarios. Firstly, where as we'll see later on, the centromere-proximal regions are very suppressed for recombination. By unlocking that suppression, it may be possible to accelerate integration. Also, using approaches that increase recombination genome-wide to create these hyper-recombination lines that will just increase levels of crossover throughout the genome. And then also, what's really one of the holy grails of this field is to have one of these very targeted recombination systems whereby precision integrations could be achieved very quickly. I'm not going to talk too much about these specific applications, but it's again something we might return to in the questions. But just to highlight, why understanding recombination I think is important, because it could lead to these kind of applications. Now, we know a great deal about the recombination pathway, and some of that is kind of summarized on this slide. And so what you can see moving down the center of this slide are 2 homologous chromosomes colored in red and blue. And this whole process begins, as I mentioned earlier, by these potentially quite dangerous DNA breaks being introduced into the chromosome. And generally what's true is that there's a much larger number of these breaks that are introduced than finally mature into crossovers. And the complex that makes these breaks is called SPO11, SPO11 complex, and you can see that represented here by these blue dots. And following that, basically a bit of the DNA becomes single-stranded and invades the homolog. And then a whole suite of genes that broadly we call the class 1 or ZMM pathway acts to cause maturation of this structure into a double holiday junction, which ultimately can be resolved as a crossover. And so this pathway down the middle is the kind of major pathway that's promoting repair of these breaks as crossovers. We also know that there's a whole plethora of different activities that repress this, that's responsible for only 200 of these becoming crossovers. So these are very interesting genes. Potentially by removing them, you can increase crossing over by kind of releasing repression. And then mainly what I'll be talking about today is also the fact that this recombination occurs in the context of a very specific chromosome structure where the chromatin is held out in these loops. There's a central, what we call an axis, that becomes something called the synaptonemal complex, the SC. And then also we know that chromatin is having a big effect, which is present throughout these loops, on how this pathway proceeds. So as I mentioned, one thing that I'll do at a number of points is to compare explicitly the wheat genome and the arabidopsis genome. And just to do that, to begin with, here we have a map of the wheat genome. And then on the same physical scale, we have the arabidopsis chromosomes up here. So you can see here that the 5 arabidopsis chromosomes kind of fit easily onto, really any of these, just a single arm of any of these wheat chromosomes. And all of the wheat work that I'll talk about was done under a BBSLC SLOLA grant. I'll highlight some contributions from the partners as we go along. And also we made use of, throughout this analysis, a very nice crossover data set released with some of the wheat genome papers generated by Pierre Sordile's lab. So a Chinese spring times Renan population where we can extract crossover data and make comparisons with this metric centimorgans per megabase, so crossovers per physical distance. And so on each of these wheat chromosomes, you can see the red is showing this recombination rate. In black is a kind of ratio of a euchromatic and a heterochromatic mark. And then in the blue here is a very specific class of transposon that is known to be enriched in the centromere. And so what you can see in each case is that you get this very pronounced smile along each of the wheat chromosomes where the recombination is very pushed out towards the ends of the telomeres. And to some extent, this is correlating with patterns of chromatin. Now, this is again just one of these comparison slides with arabidopsis. So this is now blowing up this tiny region. So again, we have the 5 chromosomes of arabidopsis now, but visualized on a much more zoomed in scale. And what this is looking at is again crossover landscape. So what would have been labeled in red in the previous slide, and that's shown by these solid lines. And so what you can see, these dotted lines are showing the position of the centromere. And you can see in all cases, there is quite a strong zone of suppression around the centromere, but it's nowhere near as pronounced as what you see in wheat. So there's much bigger genome, and this is common with many plants. The recombination is very pushed out, and actually most of the chromosome is experiencing very low levels of crossover. In a genome like arabidopsis, that's much less the case. You can see there's lots of recombination happening throughout the chromosome arms, and it's only really these quite narrow regions around the centromeres that are not experiencing anything. But you can see that that is very shared in each of these 4 crosses that differ in one of the parents. So this is an interesting experiment actually looking at the influence that different parents have on this crossover landscape. So what we wanted to do was to have a closer look at in wheat and to kind of profile some of these different proteins and kind of compare it to that recombination data from Pierre's group. And so the 2 proteins that we decided to study were DMC1, so you can see that here as this one of these recombinases, and also ASY1, which is part of, as I mentioned before, this axial structure that kind of polymerizes between the homologous chromosomes. And so the way we did this was to do ChIP-seq. So the antibodies came from James Higgins and Chris Franklin's labs, and Dan Holland, a student in the group, used those to perform ChIP-seq. And so the ASY1 data is shown at the top here by this purple shading. And on the y-axis, we have log2 ChIP over a kind of control. So it's looking at relative enrichment of the DNA that we purified associated with ASY1 as you move along the chromosome. And so if you remember, this is an axis protein, so it's kind of forming along the middle of the chromosomes. But you can see that we got this very striking pattern and very high levels of enrichment in the distal regions that broadly correlates with where this activity in crossovers occurs. Also, some public data was included here, CENH3, which again marks this centromeric region, and you can see again these cereber LTR transposons are very enriched there. So basically a very clear polarity over the wheat chromosomes in terms of crossovers, but also being reflected in ASY1. Now in the lower panel, you can see the equivalent experiment, but now done with DMC1 in green. And you can see we got a very similar pattern, that the DMC1 was very similar to the ASY1. The crossover is very enriched in the distal regions. Now, something else that I'll talk about a little bit later is that in the wheat genome, you get also something that's been noted by a number of studies, quite polarized stratification of different types or classes of genes. And so in the green line at the bottom here, you can see genes classified as having a defense response annotation. And you can see a kind of remarkable correlation with where those genes are, where the recombination is, and where the DMC1 and the ASY1 is. Now, if you did the equivalent analysis in arabidopsis, you just really wouldn't see this. So in these very large genomes, it seems that there is really a lot of stratification going on, both in terms of chromatin, but also the types of genes that you find along these chromosomes. Now, here's another comparison with arabidopsis. So again, the 5 chromosomes are here. You can see in red the crossover data, and like the crossover, and then in black here we have a marker of the DNA double-strand breaks. It's to do with SPO11. So essentially at a similar stage to DMC1 that we saw in wheat. And again, just like the crossovers, we don't see this very distal skew. It's actually the signal for both of these data sets is quite high through the chromosome arms, and in both of them it kind of plummets as you approach the centromere. We've also mapped ASY1 using ChIP-seq in arabidopsis. And again, it's quite a different picture to what we see in wheat. So you'll remember here the ASY1 is very distally enriched in this ChIP experiment. But you can see the opposite is true in arabidopsis. We actually get stronger enrichment in the centromeres. So just to kind of illustrate, I think, the importance of studying wheat directly for these things is that genome architecture really varies in plants, and although all of these players are conserved and doing different things, how they are distributed on the genome is actually quite different. This was some additional data that kind of supported the ChIP experiment. This was collected by very talented cytogeneticist Kim Osman, working with Eugenio Sanchez, that should say Moran, sorry, and Chris Franklin. Again, this was through the S. Lola grant I mentioned. And what Kim has done here is to take myocytes of bread wheat and stain them for ASY1. And in this top panel, you can see that they've also been immunostained for ASY1, and then a fish stain has been applied for the telomeres. And just like we see through the ChIP data, you can see that this telomeric bouquet, so the ends of the telomeres are kind of clustering together. You're seeing again the cytological manifestation of high enrichment of ASY1, which we think is reflected in that distal enrichment we saw through the ChIP-seq experiment. And this is also evident again with the other marker we looked at, DMC1. So you can see here again this kind of clustered region of telomeres that has high ASY1. Through immunofluorescence, you can also see there's lots higher enrichment of the DMC1 signal here. So this was kind of just a very nice cytological validation of what we were seeing from the ChIP-seq. Now, this is essentially just something that I've shown before, but now showing the group 3 chromosomes, so 3A, 3B, and 3D. And you can see for these features, which I've already described, a very similar pattern occurring over those three chromosomes. And we went on to look at basically a group of other chromatin modifications that were of interest. These two gave a very striking pattern. So these are two marks that I'll come back to later: histone 3, lysine 27 mono and trimethylation. And you can see that they gave this very kind of striking mirror image of one another. So these distal regions, which we know where most of the crossover is happening, seem to kind of undergo this kind of switch in chromatin state, where this, what would be thought of as a heterochromatic mark, the K27 monomethylation, becomes very depleted and instead becomes replaced by this alternative mark, K27 trimethylation. And a similar thing is evident comparing, again, a euchromatic mark, H3K4 trimethyl, with this heterochromatic mark, H3K9 dimethyl. And so you can see at this scale, like we're seeing very clear coordination between the crossovers being distalized, the recombinases, DMC1, and also the axis protein being very enriched in those same regions, and also these kind of broad scale differences in chromatin. Plus, again, as I mentioned, quite clear enrichments of particular gene classes, such as defense response genes here, or also the resistance gene class, the NLR genes, shown in this plot. So, the wheat genome of it on this scale, it's highly stratified to a degree that is just simply not present in genomes like arabidopsis. This is just kind of heat map analysis looking at the relationships between all of these things. You can see here this kind of, so the red shading is indicating that two particular factors, in this case, for example, DMC1 and ASY1, have a strong correlation together. And you can see here we're getting a strong correlation between these features, recombination rate, and this whole slew of what would be considered euchromatic or kind of gene-associated histone marks. In contrast, when we start to look at these heterochromatic modifications, so things that you would typically see on transposons, you can see now the relationship becomes very negative. Having said that, when we look at the relationship between recombination and particular types of transposon, it's actually quite, there's certainly differentiation there too. So, for example, the Gypsy class are very anti-correlated with where recombination is happening, but there are other types of transposons, particularly these DNA class elements like Mariner and Mutator, that actually seems to quite prefer to integrate or persist in regions of the chromosome that are recombining a lot more. So it's a relatively complicated picture there. Now, in the next part of the talk, what I'll do is to kind of switch to looking at a bit more finer scale, rather than this very zoomed out picture of the whole chromosome. And again, to compare back to work we did in arabidopsis, this is, one of the kind of take-home messages from that work was that when we mapped recombination, there are clearly what we think of as hotspots, so narrow regions of the genome that are experiencing much higher levels of recombination than other parts. And most of these hotspots, it turns out, tend to be very closely associated with genes. And what you can see in this plot is kind of analysis around genes. And the black line here is, again, looking at this data set that is called the SPO11 oligos. You can think of these again as markers of the double-strand breaks that initiate a crossover. And what that's compared to, for example, is this nucleosomal data, so looking at nucleosome occupancy. And as is well appreciated, as you move across genes, there are kind of marked differences in where the nucleosomes tend to occupy. So you can see that within this coding region, there's much higher nucleosome occupancy and nucleosome depletion in the promoter and the terminator. And what I hope you can also appreciate is that clearly the recombination signal in this case is very enriched in the 5' and the 3' regions of these genes, essentially in the promoter and the terminator. And again, underneath here, you can see, for example, one of these classical heterochromatic marks, H3K4 trimethylation, that we find very enriched at the 5' end of genes. So given these patterns, and these patterns actually have kind of emerged in other plant species as well, we were interested to kind of see whether we could have a look at the finer scale using the data that we had in wheat. And so what Andy did here was to define, using our ChIP data, kind of local hotspots or peaks, where we're seeing a kind of pile up of reads of either the ASY1 or the DMC1 ChIP-seq data. And you can see here, this green shading is kind of showing where those peaks were found in the case of DMC1 or ASY1. And the first thing that I hope you can kind of see quite clearly is that these peaks are actually much more widely distributed along the chromosomes compared to Pierre's recombination data. So all the crossing over is kind of happening out in these distal regions, but there's although it is reduced, there are many of these DMC1 and ASY1 peaks kind of throughout the length of the chromosome. And so for the analysis that I'll move on to in a second, what Andy did was to take all of the peaks, the DMC1 or the ASY1 peaks, and split them into 4 groups according to the kind of recombination rate of the region where they exist. So for example, this Q1 set of peaks is showing a higher recombination rate than these other ones. And as a control, there are just these kind of peaks just randomly assigned into 4 groups. Just before I go further with that, this kind of observation that there's potentially a lot of peaks, in this case of a recombinase, right across the chromosome where we're not seeing much crossover. I think actually, at first we were a bit surprised by that, but this is some data from Anthony Hall's group from 2019. And what they did was to map essentially evidence for gene conversions in the wheat genome. Now, gene conversion is one of the alternative repair outcomes. If you're going to repair a double-strand break, but it's not going to become a crossover, in some cases you'll see it as a gene conversion. And the key thing that was reported in this study is that actually these conversion events are quite widely distributed along the chromosome, certainly much more so than the crossover data shown in blue here. And in that paper, they also, James Higgins' group, generated these immunos looking again at a marker of DSBs. This is a particular histone variant, gamma H2AX, that accumulates at DSB sites. And it actually, is showing quite a widespread and quite a high number of foci, apparently throughout the kind of nucleus. So we think, maybe what this is telling us is there is actually a lot of initiation of recombination and repair going on along the entire length of the chromosome, but there's something special that really allows crossovers to happen just in these distal regions. So again, taking these peaks, Andy was able to look at, other features of the genome with which they kind of strongly overlap or don't overlap. And again, a lot of the similar trends popped out. There was strong positive associations with Chromatin, and then negative associations with particular classes of transposons, but not others. So yeah, so there's some interesting trends there, but what he was also interested to do was to basically go back to these groups and say, well, these DMC1 peaks are associated with crossover. What is it that distinguishes them from peaks in this, say, Q4 group? And so what these plots is showing, it's looking at different factors in these peaks, in these 4 groups around the peaks themselves. So you can see here around these DMC1 peaks, obviously we get strong DMC1 enrichment. But between these 4 groups, there's not much difference in how much DMC1 is there. And similarly for ASY1, maybe a very weak enrichment in the Q1. And a similar story when this analysis was repeated for the ASY1 peaks. But something that really stood out as kind of a strong differentiator was this histone modification, histone 3 lysine 27 trimethylation. So the DMC1 and the ASY1 peaks that are experiencing crossover have a much higher level of enrichment of this particular histone modification that was seen to much lesser extent in the random comparison. So I'll talk a little bit more about that, Mark, in a few slides' time, but also Andy repeated this looking specifically at genes. So this was, again, attempting to kind of to see what extent the patterns from arabidopsis are conserved in wheat. And again, he divided the genes into these 4 groups according to their crossover rate based on Pierre's data. And then also you can see here a different comparison. So in this case, genes were compared according to their level, or quantiled according to their level of DMC1 and ASY1. And that division also in this case was did lead to, you can see there's a positive relationship with crossover. So within genes, actually, the levels of these factors does appear to have an effect on crossover. But again, similar pattern emerged that the gene quantiles that are experiencing highest levels of crossover also again had quite significantly higher levels of this particular histone modification, lysine 3, H3 lysine 27 trimethylation. There is quite an interesting difference here though as well. For example, the kind of fine scale pattern that we observe over the wheat genes is a bit different. So we are seeing, for example, DMC1 sort of relatively enriched in the promoter and the terminator, which would kind of parallel what we saw with the SPO11 oligos in arabidopsis. But the overall shape of kind of what we see around the wheat genes is kind of rather different. Another interesting correlate that Andy picked up here was that if you just take exomic SNPs, for example, and have a look at these genes, which are, again, have been differentiated according to crossover rate, the highly recombining genes actually had higher levels of SNPs. And also, in this case, the log2 ratio of deleterious to synonymous SNPs is also quite different between these groups. So it's sort of interesting observation because it again goes back to the potential evolutionary roles for recombination. It's a little chicken and egg to kind of disentangle these correlations, but one interpretation could be that higher levels of recombination are having effects on, firstly, increasing or maintaining higher levels of diversity, and also the spectrum of that diversity in terms of its effects on coding capacity or not. Just to go back to the K27, this was also something that Kim was able to validate cytogenetically. So this data on the left you've seen before, but these are additional spreads where, again, you can see in the green, the ASY1 is highlighting these clustered regions where you're getting a grouping of chromosomes. And for this specific histone modification that we picked out as kind of very distinguishing the crossover active component of the genome, that also is very distally enriched, or it's showing this kind of clustered pattern or enrichment, again, kind of mirroring what we've been seeing from the chromatin IP data. So what is this mark, H3K27 trimethylation? It's something many of you may have heard of. This is kind of, it's very conserved, firstly. Excuse me. So you find it in animals and plants. It regulates genes. So it doesn't tend to regulate transposons, for example. There are some very famous examples of it regulating plant genes. So in vernalization, for example, these are all arabidopsis complexes, but the VERN2 complex controls a gene called FLC that regulates flowering, and it does so by adding this mark. There's also additional related complexes that are regulating seed development, embryogenesis, and also other aspects of vegetative development. I just need to plug my camera. So it's actually, the reason we were surprised to find this is that it's typically considered to be a kind of repressive histone modification. So it's something that's silencing gene expression. And as I showed you earlier, in many genomes, the general correlation that one sees is between recombination and sort of classic euchromatic marks, which would be associated with gene activity. So it was kind of surprising to us that you would see this very strong relationship between crossovers and a repressive mark. But I think the reason it potentially makes sense is that the type of genes that are regulated by this modification tend to be environmentally responsive. Genes involved in adaptation or response to the environment or biotic stress. And potentially, you could think that those genes might be genes where the benefit of recombination would be greater. So our speculation is that coupling recombination to this specific chromatin mark might reflect, in the case of wheat, a way to direct the recombination to parts of the genome or subsets of genes that are perhaps in the process of, co-evolving with the environment in some way. But that is very speculative. However, there are suggestions in the literature that this might not just be related to wheat. It certainly doesn't appear to be the case in arabidopsis, but this is data from a human study where they looked in the Icelandic population, lots and lots of crossovers in lots of families, and then correlated those patterns with various things. And notably, H3K27 trimethylation did again come out as a kind of positively associated factor. I completely missed this the first time it came around, but after we started to see those effects in wheat, it became of much greater interest. There's also some really interesting trends that Andy picked up, specifically in the NLR genes, these resistance genes. So for example, just taking NLRs again and grouping them according to how much crossover they're experiencing into these four quartiles, you could then look at within each of those how many of them, for example, are in clusters versus being a singleton, just being alone. And you can see here that the resistance or the NLR genes that are in the higher recombination zones tend to be much more clustered than the low recombination ones and have fewer singletons. There's also associations with cluster size, and then also in terms of the transcriptional behavior of these genes, whether they're induced by something called PAMPs or not. So PAMPs are pathogen-associated molecular patterns, and in this case, it's unclear why, but the NLRs that are more highly recombining seem to be less PAMP-induced. So again, and we've looked for similar patterns to these in arabidopsis over the years and not really found them. Again, I think it's kind of an interesting aspect to the wheat genome that it does show this really pronounced stratification along the kind of chromosome axis in terms of where the recombination is, how the chromatin is distributed, and also what type of genes, and even to the extent to which they're clustered is occurring. This was again just a similar analysis to before, looking in this case at ASY1 quantiles and different kind of population genetic parameters. So the first one at the top here, RM, is essentially a measure of historical recombination. It's similar to LD, but not the same. And you can see it kind of makes sense that the genes that have higher ASY1 ChIP-seq signal seem to have experienced higher levels of recombination in their history. They also have higher diversity as a measure of pi, and then also quite interesting differentiation of these statistics that look for essentially signatures of selection. So again, in wheat, quite clear patterns of differentiation with genes at the level of chromatin, what's bound there, and levels of population diversity and the signatures of selection. So, I think it's really a very fascinating genome. It's huge, as you all know, and it's very complex. But I think, from all of the work we've done in arabidopsis, I just don't think you can really understand wheat using something like arabidopsis as a model. It's just organized on a different scale. There are these very pronounced patterns of differentiation that you just really don't see to the same extent in arabidopsis. So I think it's a very fascinating genome, and I think potentially this histone modification K27 appears to be doing something quite specific and quite intimately connected with the crossovers, specifically in wheat. The only last thing I wanted to talk about again was to kind of go back to these centromeres. And something I mentioned earlier, this again is an arabidopsis chromosome, and you can see here crossovers really plummeting as you approach the centromere. This is very common to eukaryote genomes. Obviously in wheat, that zone of suppression is far greater. It's occupying most of the length of the chromosome. And again, you can see that these regions are very epigenetically differentiated. So at the bottom here, we're looking at DNA methylation, which is very much higher. And the centromeres are really fascinating. They're doing something very conserved in terms of cell chromosome segregation. But when you look between species, there's just radically different structures. So in contrast to something like the ribosome, which is changing very slowly, the centromere is changing very, very fast, despite doing a very conserved role. So this was kind of highlighted in this article as a kind of paradox. And it turns out arabidopsis has quite complex centromeres, equivalent in size and complexity to humans. And we've never been able to access these before. So recently what we've been doing is using a lot of these long read technologies like nanopore and PacBio, and that's allowed us to get complete maps within these centromeres for the first time. And as it's not particularly surprising that they're extremely repetitive, but here we've got a block of about 2 megabases of essentially the same sequence that we call CEN180 repeated again and again and again and again. And they're just fascinating regions. And once you have these new assemblies, there's lots of pre-existing data again in arabidopsis. Also, this is the case in wheat that you can look at. Now, these plots, what they're doing is looking at a variety of chromatin modifications and other features along a kind of mirrored telomere to centromere axis. And what I hope you can see is actually although it is a very much smaller genome than wheat, there is this commonality of very clear gradients as you go from the centromere to the telomere. And I suspect most plant genomes for which I've looked at, these polarities are there. And what we're excited about at the moment is being able to really look in detail at the chromatin states and the sequences in these centromeres, which we've never been able to look into before. And I think equally, it would be quite interesting to kind of go back into the wheat genome and have a much closer look at these centromere regions. It's known that they're very enriched in a particular Gypsy transposon cereber, but I suspect there's kind of more to be learned about the organization and the regulation of these regions, and also potentially how they connect to this very large zone of recombination suppression that exists around the centromeres. So I'll wrap up there, just to kind of some take-home points. The wheat genome is very, very polarized along its axis, both in terms of recombination and other aspects like ASY1, the axis. Hotspots of these factors are quite widespread along the length of the chromosome. And it seems like one of the key things that distinguishes which of these hotspots will actually experience crossovers is potentially this histone mark, H3K27 trimethylation, which, as I mentioned, is surprising, but I think it does make sense when you think about it. And that recombination in wheat is showing quite strong correlations, certainly with things like sequence diversity, the signatures of selection, and annotation, to a much greater extent than I think I've seen really in any other plant. And just a final point that, arabidopsis, as I mentioned, it's really not very adequate as a model for the wheat genome. So yeah, I'd just like to thank the people who were involved in the work. So Andy and Daniel did a great deal of the work in my group, and these are our collaborators within the BBSRC Lola program that I mentioned, particularly Kim, who did contributed those cytological pictures. And that's it. Thank you.
Thank you very much, Ian. A really fascinating presentation, and I've often been convinced that arabidopsis just didn't make sense as a good model for wheat, and I think what we're seeing now is starting to reinforce that. Of course, we can still learn a lot from arabidopsis generally about plants. So we have a lot of questions coming in, and just to remind everyone, if we don't get to your question, or Ian will be answering it in writing, and we'll make that available after the webinar. All right, well, let's dig into some of the questions. Do you think we could promote recombination in lower recombination regions in wheat, maybe with histone modifications?
Yes, I do think I think it's possible. I mean, the issue I think often is that these marks can end up doing many things. So, for example, like the heterochromatic marks are there to repress the transposons. So if one removes those marks, it could be quite deleterious, but it could also, release recombination. So I think it's just a question of finding the right combination of treatments or reductions or changes. But I think the chromatin certainly is very intimately involved in the process. So I think it is a good target to pursue. And like I say, this one of the marks that's really jumped out to us through this work is this polycomb mark.
So what is unique of the Gypsy elements to make them hide in heterochromatin? And one would imagine that transposons need expression to move, and hence would accumulate in the more relaxed chromatin space.
Yeah, yes. I mean, it's quite clear that different transposons have their own places that they like to integrate. So the Gypsies, or that general class of LTR transposon, are very often found quite close to the centromeres. The family that I highlighted, Cereber, really goes straight in, deep into the centromere, as far as we can tell. And in Arabidopsis, it's a similar picture. There's, in the centromeres that we've seen, there isn't a family called Attila. So they're the same broad family as Cereber, but quite a different subfamily, as it were. So they clearly are attracted to the centromeres. It's not clear why. Given that they're transposons, I expect they're up to no good in one way or another. But my sort of general hypothesis is maybe what they're trying to do is manipulate segregation somehow, perhaps in meiosis. And the reason, the centromere is such a complex structure is that it could be trying to kind of purge these things continually, and in doing so, it's kind of creating these repeat structures. But that is very speculative.
Yeah, a very interesting hypothesis, though.
Yeah, I think wheat. So there's so many of them in wheat that it's an interesting place to think about it. But the Cereber, I mean, it's a striking enrichment. They're really, if you didn't know where the CENH3 is, you wouldn't need to. All you would need to know is where those Cereber elements are. So they're clearly very attracted to those regions.
So you showed that the correlations between recombination rates and ChIP-seq signal across the chromosome, but there are very clear localized deviations from this. Do you have a possible explanation for that?
I think it's, recombination is very multifactorially controlled, I think. So obviously, if you have an inversion or something like that, it probably doesn't matter what the chromatin is like, because the inversion is just going to prevent formation of a crossover, or transmission of one at least. So certainly polymorphism has an effect. Also, the varying influence of chromatin and the axis, that it's, I don't think it's a single factor. Although, there are these, marks that have much stronger associations.
So the next question is, are the recombination profiles of subgenome relatives the same, or is it more pronounced in the 3 subgenome collective?
Yeah, so you mean between like A, D, and B, for example? Not enormously. I mean, the D genome tends to be a bit smaller, I guess, and for that reason, it might have a slightly higher recombination rate overall. It seems to experience probably a similar number of crossovers, at least. In terms of the other things we looked at, it didn't particularly stand out as, they're quite similar,
Now, there was one of your slides where just at first glance, you get this impression that the 3D chromosome is slightly different, really in that centromeric region. So perhaps that's consistent there. Yeah,
it could be. It could take a wheat specialist, I think, to maybe see these things. But I don't know. They are, the D definitely stands out as, the most different compared to the other two. But overall, the trends, I think, we're always there in the 3 subgenomes.
All right. Does methylation cause gene silencing every time? And what about the location of methylation?
Certainly, well, DNA methylation, if it's in the gene promoter, it certainly does silence. Many genes have DNA methylation within their open reading frame, though, and that's not at all inhibitory to expression. The K27 methylation that I mentioned, it seems like it's a threshold event. So, for example, with vernalization, it kind of, the longer the period of the cold temperature, that histone mark kind of creeps up and up until it kind of triggers like essentially a very stable silent state. And often it's kind of coating the entire gene. But it can well up in particular places, like in FLC, for example. I think it kind of initiates in one of the introns. So there probably are, certainly at the level of, say, an individual gene, like where a particular mark is, I think is relevant for sure. And also, as I showed you, recombination is also not even across genes.
All right, well, thank you very much, Ian. We've run out of time at this point. There are quite a few other excellent questions that will be answered subsequently. And I want to thank all of you who participated today and watched the webinar, and encourage you to sign up for our next webinars. And Ian, again, thank you very much for really fascinating presentation. And I'm really pleased to see you working more in wheat. That's going to be good. So thank you
very much. Thank you for your time. Okay.